Hello everyone. In this video, I'm going to explain about gene cloning. So what is gene cloning? Gene cloning is a process of producing genetically identical copies of a particular gene or DNA sequence using genetic engineering technique. So, what are the tools used in gene cloning? First, we need target DNA. So target DNA could be DNA from plant or DNA from animal. Second is cloning vector. For example, plasmid. So other than plasmid, cloning vector also have bacteriophage, YAC, yeast artificial chromosome. Okay. Tools number 3 is restriction enzyme. Restriction enzyme, for example, EcoR1, SMA1. Okay. Number 4 is modifying enzyme. Example of modifying enzyme is DNA ligase. And then tools number 5 is host cell. Host cell, usually we use bacteria. Now, what are the steps in gene cloning? There are 6 steps in gene cloning. First is isolation, cut, insertion, transformation, amplification, and screening. To remember the steps in gene cloning, you just remember ICITAS, ICITAS. So, what happened during isolation? Okay, isolation of what? Isolation, first is isolation of target DNA and plasmid. Okay, Madam terus guna contoh plasmid as the cloning vector here. And then step number 2, cutting or cleaving the target DNA and plasmid by using SAM restriction enzyme. Alright, now look at this diagram. Look at this diagram. So here we have E. coli, okay, bacteria E. coli, and we have donor cell. Now, in the first step of gene cloning, okay, we are going to isolate the plasmid. Okay, this is plasmid. Okay, this is plasmid, and this is the DNA. So during isolation, plasmid and the DNA will be isolated. So di sini plasmid dan di sini ialah DNA. This is the DNA. Alright. Now, as I said just now, in step 1, it involves isolation of plasmid and isolation of target DNA. This is target DNA. Now, in the second step involve cutting of second step is cutting the plasmid and cutting the DNA, target DNA, by using SAM restriction enzyme. Okay, so if we use EcoR1, if we use EcoR1, it will produce sticky end. If we cut by using SMA1, it will produce blunt end. Okay, now after we cut the plasmid, it will become like this. It will open like this. Okay, now when we cut the DNA, when we cut the target DNA, usually cutting the target DNA, actually we want to cut it into we want to cut the target gene. Okay, we actually want to cut the target gene. So, bila kita potong, dia akan jadi macam ni lah. Macam tu, something like this. Now, we must cut by using SAM restriction enzyme so that when we rejoin the plasmid and this plasmid and the DNA, they are complementary. Okay, now third step. In the third step of gene cloning is insertion. So we are going to insert this target gene into the plasmid. Okay, insertion. So insertion here. Okay, you're going to insert this DNA here, so it will form a structure like this. Okay, we insert it like this. This is the plasmid and this is the DNA, target gene. It will form structure like this. So actually, because this is DNA, because this is DNA, to join the DNA, actually we need an enzyme called as DNA ligase. If you still remember, the function of DNA ligase is to join the DNA. Okay, DNA ligase. Dia macam gum kan? It will form the bond here and here. Okay, to join the DNA, to join the DNA. So of course, by forming hydrogen bond and phosphodiester bond. This is the function of DNA ligase. So this is step number 3, which is insertion of target DNA, okay, into the plasmid by using DNA ligase. Alright. Now, this structure now is called as the recombinant DNA. Okay, recombinant DNA, or often called as recombinant plasmid. Okay, this is step number 3. Step number 3. Now, in step number 4 is transformation. Okay, step number 4 is transformation. What happened during this step? Now we have plasmid, a recombinant plasmid. We are going to reinsert this recombinant DNA into the host cell. Okay, ini ialah kita punya host cell. Dan kalau kamu perasan, host cell ini sebenarnya ialah ini, E. coli yang mula-mula tadi kita ambil dia punya plasmid. Okay, so inilah kita punya host cell. We are going to reinsert the recombinant DNA into the host cell. But how? How? So, saya lukiskan semula structure ini tadi. This is the recombinant DNA. Ini recombinant DNA tadi. Okay. So, how this recombinant DNA enter the host cell? So it will enter through process called as transformation, which is transformation process ini. Macam mana detail proses itu kita tidak perlu belajar sebab tidak ada dalam silibus. Okay, via transformation. Now, this is our host cell. And inilah step number 4. Number 4. Now, number 5. What will happen to the host cell? In step number 5, this host cell, this bacterial cell, are going to be cloned. Okay. Step number 5 is amplification. Amplification lebih kurang sama maksud dengan multiplication, memperbanyakkan. So, how to amplify this bacterial cell? To amplify this bacterial cell, kalau dalam lab, ini lebih kepada we culture the bacteria. Culturing the bacteria or growing the bacteria in a medium. So daripada satu bakteria dia akan jadi banyak bakteria. Banyak-banyak bakteria. Inilah kita panggil amplification. Okay, because the plasmid plasmid ini dia ada gene yang kita panggil sebagai ORI. So because of the gene ORI, maka plasmid ini boleh replicate because of the gene ORI. So macam ni lah. This is amplification. This is the 5th step in gene cloning. Okay, now step number 6. Step number 6 is screening process. Screening process. What happened during screening process? Okay, what is the purpose of doing screening? So, screening ataupun lebih tepat, lebih spesifik sebenarnya, dalam gene cloning ini kita guna satu teknik yang kita panggil sebagai blue white screening. This is a process. Screening is a process to identify the bacteria. Okay, process to identify bacteria containing the recombinant DNA. Sorry. Recombinant DNA. Alright. So, in blue-white screening, what happened is the bacteria will be cloned in a petri dish containing sugar called as X-gall and antibiotic called as ampicillin. Okay. Now, during this process, what will happen is all the bacteria containing plasmid will grow. Okay, maksudnya semua bakteria yang ada plasmid, it will grow. Kenapa? Sebab plasmid itu sendiri, okay, saya lukis sekali lagi plasmid di sini. Okay, kalau ini plasmid ini mempunyai satu gene. Ada satu gene pada plasmid yang kita panggil sebagai AMPR. Okay? This gene ini menyebabkan bakteria ini resistant towards antibiotic. Okay, ampicillin. So, kalaulah sekiranya bakteria itu mempunyai plasmid, maksudnya dia ada ini gene ini. So kalau kita letak ampicillin dalam, if we put ampicillin in this medium, the bacteria will grow because it resistant to ampicillin. Okay. Now, the problem is sama ada plasmid itu mempunyai foreign DNA ataupun tidak. Okay, mempunyai target DNA tadi ataupun tidak. So what will happen? What will happen? When the bacteria grow, okay, when the bacteria grow, macam ini, saya lukis sekali lagi di sini. When the bacteria grow, in this blue-white screening, some of the bacteria will turn into blue color. Macam ni. Okay. It will turn into blue color. Why? Kenapa jadi blue? Now, just now, we put X-gal sugar. X-gal ini sebenarnya ialah sugar. We put X-gal sugar into the medium. Okay, now, X-gal ini, this X-gal can be hydrolyzed by X-gal sugar can be hydrolyzed by enzyme called as beta galactosidase. Okay, if this X-gal hydrolyzed by this enzyme, it will turn into blue color. Okay, tapi beta galactosidase ini sebenarnya datang dari mana? Who produced the beta galactosidase? Okay, to understand this one, we have to go back to the structure of plasmid. Okay, go back to the structure of plasmid. This plasmid just now, dia bukan saja ada ampicillin gene AMPR, ampicillin resistant. Plasmid ini ada satu region, ini kita panggil sebagai, call it as, kita panggil dia sebagai MCS. MCS ini adalah multiple cloning site. Maksudnya, kalau kamu ingat tadi, in the process of cutting, okay, di sini cutting, when we cut the plasmid here, okay, actually we cut on the MCS. Okay, so plasmid itu tadi di sini, MCS ini, okay, MCS ni, multiple cloning site ini, sebenarnya berada pada satu region yang kita panggil sebagai, berada di tengah-tengah gene yang kita panggil sebagai, okay, yang kita panggil sebagai gene LAC-Z. Okay, dia betul-betul berada di sini, di tengah sini. Now, when this without target DNA ataupun target gene, kalau kita tidak ganggu LAC-Z ini, kita tidak potong di sini, tidak ada gangguan apa-apa, LAC-Z ini akan berfungsi seperti biasa. This LAC-Z will undergo transcription process, okay, and translation process to produce enzyme beta galactosidase. Ini ya, ini LAC-Z ini undergo protein synthesis and produce this enzyme beta galactosidase. So kalau ada enzim ini, dia akan menyebabkan X-gal hydrolyze, so the bacteria colony will turn into blue color. Okay, maksudnya di sini semua bakteria yang hidup ini, all the bacteria that grow here, mereka ada plasmid, tetapi dalam plasmid itu tidak ada target gene. Now, macam manakah kita tahu bakteria itu ada target gene? Okay, In this petri dish, another bacteria will grow like this. Dia warna putih saja. Macam tu, dia punya bacterial. Yang putih inilah yang mempunyai target DNA. Target DNA. Tapi, yang ini. Tapi kenapa kita cakap yang putih itu ada target DNA? Balik semula kepada proses di sini tadi. Now, plasmid ini, okay, the MCS di sini tadi, bila kita potong, when the MCS is cut, ini multiple cloning site is cut by using the restriction enzyme, it will cut open, dia terbuka macam ni, dan kita pula pergi insert target DNA di sini, target gene di sini. So what I want to say is when we cut the plasmid, actually it will interrupt the sequence of bases in the gene of LEGZ. The LEGZ is interrupted. When LEGZ interrupted, of course it cannot produce, it can no longer produce this enzyme beta-galactosidase. Okay? Bila kita ganggu ini LEGZ gene, dia punya base sequence, dia sudah tidak boleh berfungsi seperti biasa, tidak boleh produce beta-galactosidase, so cannot hydrolyze the X-gal. When the X-gal is not hydrolyzed, dia punya bacterial colony akan jadi macam ni, white color. So bila saintis kan, dia nampak macam ini, white colony ini, dia terus tahu, yang white colony ini maksudnya dia ada target gene, ada target DNA di dalam tu. Okay, so this is what we call as screening process. We want to identify which bacteria contain the recombinant DNA and which bacteria do not have the recombinant DNA. Macam tu lah. Okay, that is screening process. So itu sebabnya kenapa dia dipanggil sebagai blue-white screening. Okay, so hopefully you can understand this process. Thank you.